it just now. I had forgotten to do it. Even though we're going to focus on the Cowan PowerPoint, when we get to, or at least to amplify, you're going to see the Nestor PowerPoint the way I have it. They're both chapter 3s, and they're both under week 1 for you to review them. But I did want to mention that to you because it is, frankly, this is when it starts to get, most of this was background information and basically some organizational materials. Now we're finally getting into sort of the real material of bacterial anatomy, and this is where there's a certain degree of complexity. There's a lot of focus points, none more than gram-positive and gram-negative, and we may very well get to that, or at least introduce it in the time that we have today. So let us begin. Moving on. So all of these things are important. The DNA, we talked about that being a circle, the lack of the nucleus, those histones, if you don't remember, are in our pairs of DNA. They're proteins that kind of hold it, the spring-like shape, so it doesn't go into coiling. They don't have that. Makeup of their cell wall, peptidoglycan and other chemicals, and the fact that it has lax membrane outer organelles. So basic stuff. Why is it a living thing? Cytoplasmic membrane, cytoplasm, ribosomes, which are organelles. And again, they're the 70S ribosomes, a rather skimpy cytoskeleton. There's not a whole lot going on. There's maybe some underpinnings to sort of secure the membrane and maintain its shape. There's not a whole lot you have to know about the cytoskeleton. And basically the chromosomal structure, which we will deal with. The big players here, when we really, classification is based on the cell wall, and all cells, ours, have something called the glycocalyx, which literally means sugar shell. And that's the combination of proteins with short chain sugars that project from them. And interestingly, in bacteria, as you will see, they come in a variety of different ways that help them either hold together or they're very extensive. And there's a lot of classification and a lot of implication, particularly in ones that develop a thick shell of either sugar or a particular waxy material that we call a capsule. Because in this case, capsule is a really large structure, and it makes it very difficult for our immune system to attack. And indeed, that becomes a very significant, when we get down the road to respiratory diseases, a big, big player. So other things that bacteria have, these are appendages. Now, in A and P, you're talking about human cells, flagella. You probably studied the flagella. You looked at the old 9+2 model. We're actually going to review that. Those 9 doublets of things called microtubules might be ringing a bell with you. These are totally different. The only flagellated cell in us is a sperm cell. Okay, we don't have any pili or fimbriae. They're all much smaller, and unlike those cells, again, a flagella in our cell, a cilia in our cell, is basically a protein that protrudes and surrounded by membrane. I usually have, I even have it with, I do. Don't leave home without it.
Poop bag.
Of course. You have a dog, you have a poop bag. Threatening to bring in the dog. And it's basically you have a protein appendage that pushes on the cell membrane, and the cell membrane surrounds it and invests it. So it's really that protein is inside the cell. Now, in bacteria, a flagella, a pili, or a fimbriae in a bacteria, it's outside of the cell. The membrane's here, and its protrusion, and we base it on its construction materials, its length, and what it does. We're going to get to all that. Yeah, standard outer membrane is, let me rephrase that. It has a cell membrane, only a limited number of cells, not limited is the wrong word, a substantial number of cells have an outer membrane. We'll get to that when we talk about gram-negative. Nanowires, nanotubes, not a big deal for us. I talked a little bit about plasmids. In addition to the major genetic material, the nucleoid, or what we call the bacterial genome, many bacteria have plasmids, small circles of DNA that are classified as non-essential. Again, when we talk about essential, it means it has to have it to survive. So plasmids in a harsh environment might develop. If the environment changes, they might go away. Plasmids are helpful, but not essential. They can survive without them, and they're the ones we talk about with genetic engineering and things like that. Okay, inclusions, something we use in the lab to identify them. Sometimes there's little crystalline structures, there's sugar-like structures, there's metallic structures. Bacteria are kind of unique in that regard. So in the lab, you use those for identification. We'll mention them in just about as much shift as you're going to get them from me. Okay. Endospores. A whole lot about endospores when we talked about Pasteur and Koch and botulism and tetanus. Endospores are important because effectively they're indestructible. And there's a very good animation and there's very good information. They're really important. So one of the focus points, in addition to gram-positive and gram-negative, endospores. I'm not so big about microcompartments, and because in the archaeal as well, we really don't spend a lot of time with it. It's a bad illustration for this kind of a presentation because you can't read it. Okay, and if you want to, and we're going to go over all these different things, but effectively, this is a flagellum. It's protruding. It's quite long, proportionate to the bacteria. Much smaller and less long, less lengthy than in a flagellum. You know, remarkably small. So with that, this would be an inclusion. This would be another inclusion. Here's the DNA. Basically, the other than those little inclusions in the circular plasma here, you've got a whole bunch of ribosomes. And then you have layers. Membrane, I think that's cell wall. You may here have an outer membrane, it's gram-negative. You might have a capsule or some other kind of enclosure. And these are like little either fimbriae or pili. Fimbriae are typically smaller, they're like little bristles. So again, if you like in the fall when you go walk in the woods, you know, on your sweater you get one of those things that kind of sticks to it. You know what I'm talking about? It's little circular things, little burrs, and so and that's very often how bacteria attach to ours. Pili are not pictured here. Flagella are long, fimbriae are short. Pili, and sometimes people call fimbriae pili, have a pretty unique role having to do with genetic material being transferred, you know, really important in our genetics unit. Less so in here, but we'll talk about it. And so you can read through that, but I'm going to go over nearly all of this stuff that's there. Independent, unicellular organism. They do a lot of good and bad when they colonize. What do they do bad? When groups of bacteria colonize, that glycocalyx, if it's not organized and if it's a little sticky, you get lots of organisms that come together. They call it a biofilm. We all have a biofilm inside of our mouths. It's called dental tartar. And dental tartar isn't one bacteria, but multiple bacteria. And they play a pivotal role because of, and we really look at biofilms a lot today in their usage. That was the whole oil spills thing. Biofilms, because what does dental tartar do to you? Dental tartar sort of paves the way for specific types of anaerobic bacteria. So they're on your teeth. And so between your tooth here and the biofilm here, oxygen can't get in. We call it an anaerobic environment. And that really facilitates what are called karyogenic bacteria, meaning cavity-causing bacteria, to get in there. So that's why we use tartar agents that remove tartar, floss. We get our teeth cleaned X amount of time frequently, depending on how much you're into your dental hygiene. So many bacteria function independently. Colonies in biofilms, you're going to see colonies in the lab. There's little communication tubes, that's fine. So it's not as though they're existing, there's a network kind of an effect that's there. Very tiny, 1 micron. A red blood cell, something that you'll probably look at in the laboratory, something you'll cover when you do AP2 or have covered in AP2. They're about 7 or 8 microns. So a bacteria is, 10, 15% the size, typically, of a red blood cell. Here, a cocci, which is a shape we're going to look at, is a little sphere. It's so small that its circumference going around this little sphere is a micron. Rods, maybe a touch, fairly small, touch bigger. And then you were going to talk a little bit about pleomorphs. And they're interesting, and they do affect us a lot. Pleomorphs mean, pleo kind of implies different sizes and shapes. Morph is more about shape than size. And it depends on atmospheric conditions. Some of them are very significant too. Shape number 1. Did you see any? You start looking at bacteria under the microscope? Okay. These are cocci. Cocci are spheres, not always perfect spheres. Sometimes they look like coffee bean shaped or other shapes like that. This is a classic photo of Staphylococcus. So a coccus being the singular, spherical or ball shaped. And again, they get a lot of different variations. Here's a magnification of 10,000 times, that's 10 times more than what you would see under oil immersion microscopy. And they're clustered together. We'll get to why they're clustered together. Staphylo means cluster of. What means? A cell that is a rod. And notice we use the term bacillus. We use a small b to designate shape. We talked about anthrax, the genus of the family with anthrax, something that you're going to use in the lab called Bacillus subtilis, is a capital B and italicized. There's a lot of variation in rods. They can be long and slender. They can be what you would call dumbbell shaped. They can be kind of rounded. We call those coccobacillus. We can chain them together. There's lots of variations. So as you can see, blocky, spindle shape, long threaded, club shape, whatever you want. And so, and you'll see coccobacilli. You'll work with one called Enterobacter aerogenes in the lab. That's more of a coccobacillus. Here's a very, very famous rod-shaped bacillus, Legionella pneumophilia. Anybody ever hear of a thing called Legionnaire's disease? No? Yes? Oh, anecdote will be coming when we cover respiratory diseases. I was there. First came to our attention, I know, during the bicentennial, 1976, okay, in Philadelphia. American Legionnaires gathered in an old, anybody familiar with the Rittenhouse Square area of Philadelphia? It's right in the middle of downtown. It's an older area. It's there. It's not the historic area. It's a little, just not too far away from it. And they had an old hotel that kind of reminds us of our William Penn. Okay, older hotel, that vintage, called the Bellevue Stratford. Probably under a different name. And the American Legionnaires gathered there for the bicentennial. And these were, we even had World War I vets still alive back at that time. People who were 75 to 80 years old. World War I, World War II, Korea, Vietnam era veterans still there. And when you're in a convention like that, you're always in the same rooms. They just partition them for lectures, partition them for dinner, partition them to sit around and talk afterwards. Everybody had a history of smoking and drinking, just about. Pretty common. So about 76, you could do all that stuff. Right? And about 15 died. And they thought it was an outbreak of the Spanish flu because some of them were alive during this deadly flu stage. And you remember I told you about me looking at viruses. I was taught by the lead virologist in the Commonwealth of Pennsylvania. They put him up to identifying the virus. He could not because he didn't know what he was doing. It was a bacteria. It's a bacteria that only grows in dusty, wet, humid areas, the old crawl spaces in old buildings. We'll get more into that. We used to have our, honor society meetings at that hotel, probably in the same set of rooms. You're going to find, there's a lot of, I was there. You're going to come. Yes.
You were too young.
Yeah, I was just a twinkle in my parents' eye. So, shapes. We have the basic shapes are cocci, rods, spirals, which they'll get through, and more or less curved forms. So Vibrio, very famous because of cholera, it's named Vibrio cholera, is a slightly curved or comma-shaped bacteria. Some of them are gentle spiral forms. Some are more, you'll see called spirochetes. When we get to digestive disorders, Campylobacter jejuni, because they find it in the small intestine, the jejunum, is one that has this sort of a form like that. And then lastly, spirochetes. Spirochetes look like a really tight spiral, like a spring that you would see. Okay. Very much spring shaped and very, very famous. Two major diseases, one called syphilis, which I talked about briefly before, was part of that. It's all right, the boss is texting me. I am by forced to respond. Hey. If I don't do this, I'm in big trouble. You didn't get back to me. Okay, and the other disease, very famous around these parts, Lyme disease, caused by spirochete. You're going to see something very interesting about spirochetal diseases is they particularly go through a series of phases that are quite notable. And then we have some oddball ones. This is one that has, it almost looks like a fungus, these filamentous types, and sort of little new bacteria come off. This is called streptomyces. A lot of the antibiotics come from this family. A lot of antibiotics. Endospore forming bacteria, and these filamentous bacteria have a lot to do with antibiotics. Okay, how they're arranged. They're not necessarily, some of them are all just individual. E. coli doesn't have little specific things. It's just a generic, it's a prototypical rod-shaped bacteria. We see them, it depends on how they replicate. If they replicate in one plane, we see pairs or chains. If they replicate very symmetrically, we see groupings, okay, cubical packets. And if they just or irregularly replicate, we see clusters. So you see the name streptococci for chains. Pairs would be diplococci. And a lot of times you're going to hear, we'll talk about streptococcus pneumonia and diplococcus pneumonia and pneumococcus. They're all the same bacteria. They're the leading cause of bacterial pneumonia. But we've taken to naming, even though they're the same, naming them, because sometimes they'll be in chains, sometimes they'll be in pairs. Staphylococci or something called micrococci, which you work with in the lab, form those clusters that you saw before. So here you can see if they have one plane, pairs or chains, little packets, we don't see so much, and then these irregular shaped cluster, staphylococci. This is a very unique form, and when I did the lab, I used to get this stuff. It's called Corynebacterium. We have a ton of them on our skin and our respiratory they're not bad guys. Some of them are bad guys. They form palisades. And so what happens is when they replicate, the new one kind of sticks to the old one. And in the old days, now they call it a palisading pattern because it was pejorative. They called it a Chinese letter pattern because it looked like Chinese characters, like the calligraphy characters, which I always wanted to figure out what they meant. Okay. And so the very, very specific, you're going to see some of them You did environmental samples this week or last week?
Did blood
Okay.
microscope. We just started it. We're doing that next week.
Okay. When you do environmental samples, you might find some of those. Some of the environmental samples.
We took the swabs for it on Monday.
Yeah, like bottoms of the shoes and
Yes.
stuff like that.
Yeah.
Good stuff. You'll take off your shoes before you get into the house, even if it's freezing. When I used to do that, my students would never use a water fountain again. When you put your mouth on it, they're gross. You're disgusting. Hey, can I borrow your phone? Not on a bet. Unless you're texting somebody. I don't want to talk in that thing. Moving on. And the rest of those, okay. Moving on. Appendages. Sticking out. Flagella and axial filaments. The difference is flagella are neat. They are connected and attached and they protrude. They're big. You're about moving around. Not all bacteria are motile. In the laboratory, most likely you will inoculate tubes and look on plates to see if they're, because it's how, look, everybody. Not that many of you are going to do that, but the whole ball game to treat somebody with a bacterial infection is what bacteria is it? Well, we've got to be able to identify and isolate it. And it's like what I call biological barcode. Okay? Is it gram-positive or gram-negative? Is it a rod or a coxon? It's almost like a yes-no type of thing. Thick line or a thin line that you would when you scan a label at the store. You're doing self-checking. Biological barcode. These are the types of things. Does it have endospores or doesn't? Is it aerobic or anaerobic? It's very much of a yes-no kind of thing. So we look at these types of flagella are another one. Does it have flagella? Does it move a lot? And if so, how are the flagella arranged? You can stain them to find them. I don't because the stains are nasty and they smell bad. And you normally have to do that under the hood in the laboratory. So you have the filament and it's different. Our flagella just go back and forth like this. It's like a whip-like motion. Bacterial flagella are like a helicopter. They can go this way or that way, depending on which direction. So the filament, it has a hook and it has a basal body that attaches it. So they're showing it to you here. We'll get to the cell layers down the road. It has the basal body here, kind of proteins within the membrane. It's got the hook that kind of takes it away from the cell. And as you can see, depending on the energy, it can spin one way or the other way. And that's kind of how bacteria get around, the ones that do. A lot of them will just move in the water or move in the air. I mean, if you walk into the room, bacteria is moving around. Step on the floor, it's moving around. That type of stuff. But you can see the way it looks here. So we classify it based on that. And it has to depend on the arrangement. For instance, that one you saw initially had one long flagella. If it's single, trichus because they look like hairs. Okay, monotrichus, one hair. If you have a bunch, lopho. If you have one on each end, like ambidextrous, amphitrichus. And the ones that move the most, okay, you're going to see one of these in the lab called Proteus vulgaris. Peritrichus. Peri for around. So know your root word trichus, mono, lopho, you just have to remember. Amphi, I think ambidextrous, like that. Late Yogi Berra, so half amphibious. No idea what I'm talking about. Yogi Berra, who was named for kind of, I'm trying to remember historically, Lawrence Peter Berra from St. Louis, one of the great, all-time great catchers, Hall of Famer from New York Yankees. Growing up, I'm a Yankee fan. So sooner or later, we'll win again. I'm also a Knick fan. We haven't won in 50 years, I'm hoping. Flagella are dispersed randomly. So here they are, mono. And I mean, I don't know, it's hard to tell from that one. This would be more like a tufted one. And you'll see the arrangement. You just have to know what they are. that are there. And there are probably some better illustrations. And what makes them move? Bacteria move because of environmental changes. So there are basically 5 things that permit them to move in no specific order. But surely the most common is this, chemicals. So bacteria will move to chemicals, and it'll either be positive toward the chemicals, like being attracted, so the propeller spins one way, or repelled, the propeller spins the other. So positive and negative chemotaxis. Some move to light, phototaxis, I'll just do that. So, oddball bacteria actually move to magnetic influence, magnetotaxis, heat, thermotaxis, and even air. Bacteria move close to the air, like in a tube, as you use in the laboratory. If they're aerobic, they'll be at the top. If they're more likely anaerobic, they'd be at the bottom. So aerotaxis. So there's lots of different ways. And that's how we talked about living things respond to their environment. That's how indeed they respond. So, and they don't do it directly. Bacteria have limited energy resources. Most bacteria ferment. Fermentation means you make about 2 ATPs versus what we do, which makes 36 to 38 ATPs per unit of glucose or whatever sugar substrate. So they don't make a lot. They've got to be very energy efficient. So you and I, if we wanted to walk to the store, we walk back here. Bacteria do run, tumble, run. They make a run. They use up energy. They tumble while they're regenerating their energy, and then they make another. Eventually, they get there. It doesn't always go in one direction. They probably have some illustrations and surely an animation about this. So they'll go in a straight line, and when they stop, they'll tumble. And again, some of these a little bit more differently than others. So it's not straight. It's not like you and I or something from one of our cells moving in a specific direction. When you're injured and a white blood cell is going to the site of an injury, it goes fairly directly to that injury. Be the way to describe it. It dedicates its energy resources to that. So that's what I mean. So this is, again, starts here, ends here. Certainly not a direct line. Now, some bacteria just move randomly if there's no attractant or repellent in there, but whatever the source is. So if the environment is unchanged, it'll be moving around. And so we look at motility as one of the things, whether or not it moves or not, and to what degree is a way to identify it. To me, it's like the overlap here between lecture and lab. So I mean, it's just not feasible when we have whatever 3 big microsections and 6 labs, whatever we have, to have the same lecturer and instructor. It's just too many hours that you have to do. So so I have a pretty good idea of what they do. I've done the lab here, I'll tell you. Spirochetes are different. Okay, they have inside the cell, they have threads that are spiraled. They call them periplasmic flagella or axial filaments. And you have to, it's easier for me to show them to you if I don't know if they have it. I don't see it. So I guess the best way to, to sort of tell you about it is they're spiraled to start with. They have little filaments inside, not outside of the cell, and those move. It's kind of like doing this. Okay, so I think of a really good analogy to do that. In surgery, we use something called a jiggly saw. When you have to saw through a bone. Most days now we do it with a power tool. In the old days, it was like a back and forth thing. It's like if you were in camp or in Boy Scouts or Girl Scouts, and you have like a stick and you'd be pulling back and forth, rubbing 2 sticks together. That's kind of the way they move. So the movement is inside. You don't have anything protruding, it's inside. It's in an area called periplasm, which we'll talk about in gram negatives. Here. Yeah, because I remember when we were doing in surgery, a lot of times we used that as opposed to using the power tools. Usually we use nitrogen power tools because we use nitrogen because it's not explosive in surgery. If you use oxygen or something else, friction, boom, bad. Never bad to tell the patient their leg exploded. Fimbriae, bristle-like fibers, and that's how they attach. So this would be relevant to you ate something that probably had too much, that you wasn't pasteurized properly. You got sick to your stomach. Those fimbriae are how it adheres to receptors. It's all about it. Bacteria and infections are very specific. I mean, I'm not telling you to do this, but if you took bacteria that would cause you to have a stomach ache and put your hands in it, assuming your skin was intact, it'd be no problem. You know, it's all about receptors and things like that. You know, you can stick your hand into something, and unless it's something that attaches to your skin, theoretically it wouldn't attach. That's why it's so important when we talk about it when it comes to hand care. You're going to be working around people, little nicks and cuts in your skin, because they're passageways for you and they're passageways for them to get hurt. But this is what happens. It has to be specific. That's why hand washing is so important. Because when you're working around people, they have all sorts of stuff on them. Okay? Skin's not sterile. Pili, flagella are long, fimbriae are short, pili are in the middle. And what they do is it's kind of a neat process called conjugation. They used to think they'd have bacteria that had projection. Bacteria didn't. When they put them together, they would kind of look like this. And you'll see when we do our genetics units, they talked about this being male bacteria and this being female. Because erroneously they thought that this was some kind of sexual congress. It's not. The pilus is a hollow protein tube made out of something called pilus. I have this on handouts for this. Flagella, the repeat unit, is called flagellin, and for pili, the repeat unit protein is called pilin. So it's not anything similar to what we have in our cells whatsoever. So the reality was this tube is effectively, and we have a really good animation, the genetics unit, that basically allows genetic material to transfer from one cell to the other. It's the remarkable thing about bacteria. You and I, we're only experimenting with gene therapy to resequence our genes, to take a bad cell and make it into a normal cell, or a normal cell and make it into an abnormally good cell. Bacteria do that overnight. Again, if I didn't tell you, I'll just tell you it again. I mean, if you have perfect vision, you could touch her and she would have perfect vision, and everybody else, as you touched everybody, that trait would pass on to all of us and would change the population overnight. Me too. Somebody's a great athlete. I have a vertical jump of a half an inch.
Nice.
That was it.
Amazing. That's pretty good.
No rebounds. I could touch Tiger Woods and all of a sudden I could hit a golf ball 300 yards. You know, please. One day. So, well characterized. We see them more in gram negatives than gram positive, but staph can do this overnight. That's a gram positive. Genetic material, act like a fimbriae, assistant attachment, act like a flagella, make a bacterium modal. There's a lot that they can do. So they're really an important area. And these little nano means really microscopic. A nanometer is a billionth, where a millimeter, you have a millimeter is 1,000th, a micrometer is a millionth, a nanometer is a billionth. And so tiny extensions of cytoplasmic membrane, and they transfer materials. You're going to see bacteria deal with iron-rich substances a lot. And it's not like they're breathing it with what they're talking about here, certainly. But a lot of times what our liver does when we get an infection is it holds on to iron, because that keeps it away from bacteria that thrive on it. So here, and you see this all the time. Fimbriae are the little bristles that you can barely make out. Pili are the tubes, interconnected. And there's different things. Chain links in a fence, bacteria in a hostile environment. This is almost like protecting themselves. Okay. So, and again, that underscores what we didn't think about bacteria. Oh, we'll just get the most powerful antibiotic and we'll give it to somebody in the hopes that it'll get rid of their infection and they'll get better sooner. And, the doctor will be a hero. The reality is that it's a living thing. If you bombard a living thing, if you bombard us with adverse environmental conditions, we'll change, we'll adapt. We'll develop things that will help us adapt to that environment. Those will survive and they will be stronger. That's what resistance came from. That's why we begot MRSA. That's why we have potentially bacteria that we call superbugs to the point where we don't have antibiotics. We're getting to that stage. So that's just something to remember in the back of your mind. And here, this I talked about before, these sugars on the outside. So they're sugars projecting from proteins. If they're loose and poorly organized, inherently they're sticky. It's sort of like I love candy analogies, as many of you know.
I love candy.
Hey, you're preaching to the choir. I told you, that's my granddaughter. I always checked her, my oldest, who's now 22. I made sure if they got candy at Halloween, I made sure that there was nobody tampered with that candy. And she really had a run of suspicious Hershey Kisses and, different candies that I really liked. And I felt important that I test them, so I ate them.
Of course. Yeah.
Reese's Pieces. Oh, I don't like the looks of this. I don't have any. I know I got some. I thought they were tampered with. Here, take this. So, as a candy, so if you ever get like the Sour Patch Kids with the sugars on them, they're sticky. It's not like a great M&M. M&M is more like a capsule. And those other ones, particularly leave them out in a warm environment, they kind of stick together. That's the difference between a slime layer and a capsule. So here's the glycocalyx. So you can see there's a lot of coats. Cytoplasmic membrane, the cell wall, which we haven't even gotten to, the S layer for adverse environments, glycocalyx. It's not like it's just sitting there. It's hard to attack. And these we call encapsulated bacteria. You'll do this in the lab, most likely. You'll do what we call a capsule stain or a negative stain. This is a negative stain. You're basically seeing this is the stain, this is what doesn't stain. Okay, this is the bacteria. The capsule is sort of keeping the stain away from it or looking a different color. So a negative stain, it's great in the lab. It's kind of like we use India ink or something called nigrosin, and it's repelled by the bacteria, and it looks for all the world like a light bright. Like a little kid's light,
I have one of those.
We sell those. Oh, you're awful. Nobody got excited about a light bright.
I had one.
Yeah, I'm very happy with that. Let's see, what do we sell recently? We sell a lot of yarn. Yarns. Oh!
Those are one of those.
2 orders to ship. Ooh. We got,
You shopping?
No, I'm saying it's what we're selling on Amazon. A Squishmallow clip and something, I don't know if it's a DVD or not, it's called Pick of the Litter, a dogumentary.
A documentary? Are you kidding? That's hilarious.
Yes.
Did you get that on an email or?
No, yeah, it's on my phone. Use our phones. You know, you got to make a living.
Oh, you're selling stuff on Amazon.
For sure, my wife.
Oh,
I
okay.
merely assist. Okay, capsules. A lot of bad guys make capsules. Okay, the reason the capsules are bad is you can't phagocytize them. Our body's defense is based on the concept of phagocytosis, meaning we gobble up. I love that. We gobble up things that hurt us. We have macrophages, big eaters. I want to be reincarnated as a macrophage. Okay, biofilms, dental plaque. And it's a lot more significant in nursing and in medicine. IUDs, if they're still being used, pacemakers, a lot of orthopedic hardware, catheters, particularly urinary related catheters and other catheters, they get slime layers on them. They're a big problem. And when there's an infection, it's very hard to penetrate those. We only can use specific antibiotics to get through the slime layer. So this is what it looks like when you look at a biofilm. Okay, it starts with a surface, it gets colonized, the glycocalyx allows cells to stick together, additional microbes get into this sort of a film, and so you have a group of different kinds of cells. So they have, and it's almost like they have a life of their own. And the ones that are close to it are oxygen aerobic organisms. One with oxygen are up here, anaerobic organisms down here. That's the problem with dental caries or cavities. In fact, Streptococcus mutans, which is the cavity-causing bacteria, is more anaerobic. It doesn't like air. And so and now we get to, and it's well expressed. And this is the point I'm happy we got a few minutes to touch on. The best term for me to use in all good textbooks is called a cell envelope. We have a membrane. That's it. We don't have a cell wall. Plants have a cell wall. The idea of a cell wall is better to think of it, the combination of membrane and wall, as a functioning unit together. If you disrupt the membrane, you disrupt the enclosure. If you disrupt the wall, you disrupt the enclosure. Doesn't matter. They function together. It's an envelope, an enclosure. 2 or 3 basic layers, each functional, but they work as a single protective unit. They include the wall, the cell or cytoplasmic membrane, like all living things have. And they have, particularly in gram-negative bacteria, an outer membrane that's very interesting. This is the hard part that we're getting to. We'll do it on Monday, but I'll give you a little introduction. This is the complexity. If you study and stain gram-positive cells, you have a membrane, a standard membrane, embedded protein. You have multiple 20, 30, 40 layers of this magical substance, peptidoglycan, anchored to the membrane by something called teichoic acids. Inherently, this is porous, so stuff can get in and stuff can go out. Here, the only thing that's the gatekeeper of stuff going out or stuff coming in is the membrane. So you can see it here schematically, microscopically, very significant, and they're relatively easy to study. And for us, you'll see a ton of them for staph and strep, which are fairly easy for us, are there. Not so. This is, and it's total, you can see just looking at the illustration, the total difference. This has a mountain of peptidoglycan. This has comparatively little. This has a standard membrane on the inside. This has an odd membrane on the outside. They're not the same. This, phospholipid bilayer. This is a phospholipid. This is not. This pinkish color here is called a lipopolysaccharide layer, or LPS layer. It is, here's that word again, unique to gram-negative bacteria. It's something only gram-negative bacteria have in the living world. We don't have it. That's a big problem. Gram-negative bacteria go where they're not supposed to. There are big problems associated. Because to all of us, they are toxic in some way, shape, or form. And because it's still a membrane, it makes it it's not porous like layers of peptidoglycan. You've got to have, as you can see, passageways for things to get through. That's why we have a distinction between the antibiotics that we typically use for gram positives and gram negatives. You can penetrate this fairly easily, not so with this. And so that's a small snapshot of the complexity that you have to deal with and recognize. And it's being honest with you, for the first exam, this is probably the major focus point. What's in gram-positive? A lot of the questions are, what's in prokaryotic cells and eukaryotic cells? What's in gram-positive? What's in how do they differ? Or how are they similar? What do endospores do? Those are the players that honestly, that's going to be on the study guide, and I probably have it on there, and I'll send it to you, whatever I think of it. But that's what we're talking about. So that basically, cell walls are about, that's give me what I'm going to be on, are about shape and support. Let's stop here because I'll have to answer. Bye-bye. I can't take it. If I don't respond to my wife, I'm in trouble.